Welcome to another microbiology lecture. Today's lecture is going to be talking about the pGLO plasmid that we've used in lab to transform E. coli. Let's go over the details. So when we think about genetically modifying organisms, we want to go back to the dogma of molecular biology. Because what we're doing ultimately is changing, in this case, the available DNA to be red. And by changing that blueprint, we can produce RNA that then is translated into proteins, and those proteins do stuff. In the case of pGLO, we're going to be producing a protein called green fluorescent protein. So green fluorescent protein is native to a sea jelly, or jellyfish, try not to say that anymore, Equoria victoria. And this has been incredibly popular in molecular biology because it's an excellent marker protein, because you can see it, because it will fluoresce, i.e. when you excite it with UV light, it gives off a bright green color. And then you can tag it to other proteins so you can track them, or you can tag it to a certain promoter to see when that promoter is activated in the development of plants or animals or fungi or even in bacteria. So you can see here a picture of fish that have been transformed under a certain promoter with GFP, so they glow green. And then you can see at the bottom there a plate that is similar to what our final results will be in this lab, which are individual bacterial colonies that have been expressing the protein and now can glow green. So this is what GFP looks like. Again, the cool thing about GFP is that the chromophore, the component of the protein that captures light and then re-emits the green photon, is derived completely from the amino acid sequence. Right? So there's no extra stuff you have to add to it, which is pretty cool. Again, this protein has been extensively studied and tweaked. So now we have a whole family of colors that were derived from the original green fluorescent protein. On the top there, the EGF is actually enhanced green fluorescent protein, which gives a brighter color with less activation. And you have yellow and orange and red and purple and blue and in-betweens as well. So there's lots of these versions out here because, once again, what a great idea. If I can have more than one color, I can track more than one thing. And a lot of the images that you'll see of cells will be a fluorescent-based kind of localization for cellular components, and some of those can be derived from one of these versions of GFP as well. So in this lab, what we did was we took E. coli and we performed a heat shock transformation, which we'll go into more detail at the end of this lecture. But these little orange circles mean these are representing the plasmid. So we get the plasmid into the cell, and the plasmid is making beta-lactamase, which is our ampicillin resistance, and of course GFP. So first what we're going to do is we're going to look at the plasmid in detail and how we control expression, and then we'll go in and return to the details of the heat shock transformation. So again, a plasmid is just a circular piece of DNA that we find in bacteria originally, autonomously replicating, so it will maintain itself because it has an origin of replication. And then more often than not, there's some gene on that plasmid that is advantageous for the bacteria that is harboring it. In many cases, these are antibiotic resistance genes. pGLO specifically, the construct has our origin of replication, ARC, GFP, and the beta-lactamase gene. And we can represent that map of the plasmid a couple different ways. So you can see on the left a more detailed map, which includes all the restriction sites that are common. If you wanted to modify this further using restriction digestion and putting new pieces in, what have you, this would be something you look at. Or we have our kind of simplified version that highlights the genes that are currently part of the pGLO plasmid. Again, beta-lactamase is going to be our ampicillin resistance. Green fluorescent protein is the Equoria victoria derived protein from that jellyfish. And then ARC is going to be our regulatory protein. Because this is a construct, we've basically cut and pasted different sequences of DNA together to kind of this amalgam, right? So this plasmid construct. And part of the way that we're going to talk about, part of the way we control expression of GFP, what we're going to talk about next, is using the arabinose promoter, and that's going to need the ARC regulatory protein for it to function. So we need to have that on the plasmid, and that's our last gene encoded by this plasmid. So gene regulation, as I said before, is going to be controlled by the arabinose promoter. So we call it the pBAD promoter. And so in a native setting, when any given bacteria has the operon to metabolize arabinose, you would see control of the B, A, and D genes respectively. Those are all the genes, the proteins that the bacteria needs to break down arabinose and feed it into metabolism so that you can get energy out of it. Those are the three, that's the machinery it needs. And so again, thinking about control of gene expression, when I don't need that machinery, I don't express that machinery. So one way to be sure I only express it when I need it is to make the target of that machinery, in this case arabinose, a sugar, to be the effector, or this in the case of the inducer. So what you can see here is ARC at the top on the left there is an oval shape that's kind of tipped on its side. It's binding. This represents the DNA and the operator, and then the operon containing the genes. So ARC is currently not allowing RNA polymerase to access the operon. And when the effector, when the inducer, arabinose, binds to it, you see it tips vertically. And now you create more space between the ARC protein and the genes to be expressed, and this creates opportunity for RNA polymerase to bind. ARC actually upregulates the binding of RNA polymerase, so we turn on the gene and we turn it on aggressively. So that makes sense in a native setting because now I'm going to make the machinery that I need to break down arabinose so I can use it for energy. Again, the beauty of molecular biology is that I can cut and paste. So if I take that same sequence and cut it out of the native genome, I can put it anywhere I want, and it will still work pretty much the same. So in our pGLO construct, what we've done is put that down in front of our GFP gene. So again, when arabinose is not around, the ARC protein will bind to that promoter, that sequence of DNA we find upstream of GFP, and prevent transcription. Then when arabinose is added, the ARC protein changes conformation, changes its orientation to the DNA, and then actively loads RNA polymerase. This is that same graphic in a different form, right? So again, explaining on the left how the arabinose operon works, and then on the right, how we have that same system used to control the expression of green fluorescent protein in the pGLO plasmid. So again, we're talking about using the pBAD promoter to control gene expression of the GFP protein in our pGLO plasmid. So methods of transformation. So we did not use electroporation in this lab. It is a viable and efficient way to get DNA into a cell. Rather, we used a heat shock method, which incorporates a solution of calcium chloride. We use chemically competent cells to uptake the DNA after a heat shock. And let's look at some of the details of that so we understand it a little bit better. So transformation. The transformation solution is a calcium chloride solution. I think it's like 50 millimolar, but I could be wrong, so don't hold me to that. What we're basically doing is we have calcium ions in abundance, and those positively charged calcium ions are going to bind to any DNA just because of charge. So electromagnetic attraction, force in the universe, says you're negative, I'm positive, it's going to push us together. And so you're effectively shielding much of the negative charge of that DNA molecule. What DNA molecule? We're talking about our plasmid, because the plasmid is what we're going to add then to solution with the calcium chloride and with our bacteria ready to take it up. We want to remember that, again, membranes have a hydrophobic core, and that hydrophobic core is very, very robust at preventing charged molecules from moving through it. So by reducing the kind of total charge, you're not actually reducing the charge, but apparent charge of the DNA plasmid molecule, theoretically we think this is allowing it to move through the membrane easier. So we know we have an incubation step, so we're going to incubate our cells on ice for 10 minutes, and that's going to really slow down the fluidity of the membrane. It's also going to increase the delta of the heat shock. So if I went from, the heat shock's at 42 degrees Celsius, if I went from room temperature, which depending on where you are is usually around 20 degrees Celsius, then I would have a 22 degree or about a 20 degree Celsius delta or change. But if I take those cells down to 4 degrees Celsius, which is basically water on ice, now I have a 40 degree or so swing in temperature, and that makes the shock a lot more shocking. So when I do that, I'm going to increase the permeability of the membranes by making them more fluid. We also know that the bacteria themselves are going to respond to that heat shock because that heat, 42 degrees Celsius, is higher than that bacteria is comfortable at least the bacteria that we are using. And so then it's going to respond on a metabolic level, change its gene expression. And one of the things that we found is that facilitates DNA uptake. So because we provided then in the environment the same solution, a bunch of plasmid DNA to be taken up, some of it will be taken up, right? The next step then is we're going to take those cells just for that, after that short heat shock, we're going to put them back on ice, cool them down, and then ultimately transfer them into a happy place with LB broth. Again, LB broth is not, it's just one of the rich media that we use in the lab. It's particularly good when growing E. coli. It's going to give you everything you need to grow if you're a bacterium. So your carbohydrates, your amino acids, nucleotides, salts, vitamins, all that stuff. So it's happy because I've just been heat shocked. And more importantly, I want to make sure I give time to express the genes that are going to protect me from the antibiotics. So that 10 minutes in LB allows me as an E. coli cell to grow, express the proteins that you've just given me on the plasmid, and then I make that beta-lactamase, the protein, that will then protect me when you transfer me to a plate that has ampicillin on it. So our last step then is to plate out those bacteria on 3 different environments. So LB and AMP. So now I've created a selective media by adding ampicillin. It should kill all bacteria that aren't resistant to ampicillin. A second environment is LB, AMP, and arabinose. So now I've added the inducer to our selective plate, right? So now cells that harbor the plasmid and have expressed ARC and everything is working, now arabinose should interact with ARC and allow the GFP protein to be expressed. And then, of course, we have just our standard LB plates with no ampicillin. So when we think about those 4 groups and the 4 actual plates we're plating out, you have your control group and your experimental group. So those cells that you exposed to to our pGLO plasmid should be expressing ampicillin, should be expressing our GFP. And so we should see growth on the first plate with LB and AMP. We should see growth on our second plate with LB, AMP, and arabinose. In addition, though, when we expose that plate to UV light, we should see those cells glow bright green. Whereas on the first plate, exposure to UV light should not change the appearance of the bacteria at all. Our pGLO minus plate with LB and AMP should be blank. This is a control for our ampicillin to make sure it's effective and that our plates truly are selective. And then the pGLO minus plate with just LB shows that our cells are actually growing, that they're working and they're happy under normal conditions and have kind of survived the procedure. So that's the kind of the pGLO lab in a nutshell. You should be able to describe the control of expression of GFP most importantly. If you understand the specifics of the transformation, that's a bonus for you, but I won't necessarily highlight that in assessments.